So today's talk is by Professor Tanel Tenson. He's the Professor of Antimicrobial Compound Technology at the University of Tartu. So he earned his PhD in 1997, focusing on bacterial protein synthesis and antibiotic mechanism. So after his postdoctoral research at Uppsala University, he established his own research group in 2003 with fundings from the Wellcome Trust. So Tanel has led major research centers and coordinates national programs on antimicrobial resistance, including AMR RETA project in 2019 to 2022, which focuses on monitoring and controlling resistance through One Health approach. So his expertise spans biochemistry, biophysics, system biology of antibiotics, and their impact on human and veterinary medicine. Today, he's going to talk about antibiotic against non-growing or the non-culturable bacteria. And the floor is yours.
Okay. Thank you for inviting, and nice to visit this great university. I was thinking what I will tell today, and we are working on different aspects of antibiotic action and antibiotic resistance. And I was thinking that it's the last moment that that if you're working on One Health, then I will just to highlight some of the directions that we have been having. It's a One Health approach. Now we started this from 2010, and where we are with One Health approach in addition, and then of course we have been involved in different projects on antibiotic development, also with companies and antibiotic resistance, and then in the second half I will tell you about some of our ideas how to improve antibiotics. First, what is the problem with antibiotics is that if you are looking here where the antibiotics were developed, then the first antibiotic actually was not penicillin, but this were the sulfonamides, and those were developed in 1930s. And then came also other antibiotics, you know, and for the late 1960s, it was already the idea was that we have enough antibiotics to cure all the infections that bacteria can cause. And then for a certain time period, there was no development of new antibiotics. And then when antibiotic resistance problems started to come in, and it took quite a long time to have new antibiotics coming, and here we had linezolid, daptomycin that were really new antibiotic groups, and maybe we have one more nowadays, but certainly it is really not so easy to discover new antibiotics. At the same time, if you are looking where the first clinical resistant isolates emerge, then it takes from 5 to 10 years where you start to see clinically that some bacteria are resistant. And first, it was not a problem because you have some time period where you can if there is resistance to one antibiotic, then you can choose another one, and the resistance levels were not high. And then what was the first really serious case was the emergence of methicillin-resistant Staphylococcus aureus. This occurred in the second half of 1980s. This is data from US, but the same was in other countries, and at this time only antibiotics that you could use to treat this medicine-resistant Staphylococcus aureus was vancomycin, and you were also seeing that vancomycin resistance in Staphylococcus aureus is also increasing, and if those resistances would combine, then you will have infection. You will have infections that you can not treat with antibiotics. Yeah, Staphylococcus aureus, this is something that people get quite often in the hospital. If they have a major surgery, then they get this infection quite often. So this is one of the most common hospital-acquired infection. So this is quite common and not being a problem. And this was a time where people started to talk about antibiotic resistance, but this issue didn't realize that the medicine and vancomycin resistance didn't combine, so we didn't have a secretion of the multi-resistant bacterium at this time, but nowadays, and also the new antibiotics that have been developed, they are active against Staphylococcus aureus and other gram-positive bacteria that have different cell membrane and cell wall type, and it's really harder to get new antibiotics for gram-negative bacteria that have two membranes, and the uptake to get antibiotics in is really a big problem. So now we have bigger problem with gram-negative bacteria. And what does if we are talking about the resistance that emerges? So what we see is that the bacteria get mutations in the chromosome or acquire genes that do something to antibiotics. So you can mutate the target site. Antibiotic has a target in the bacterial cell, and you can mutate the target site. Then you can pump the antibiotic out of the cell. And what else you can do to the antibiotic? You can inactivate it. So basically those are the three ways how to handle the antibiotic and how bacteria are getting resistant. And basically, from where the resistance comes from is that most of the antibiotics we are using are natural compounds. So microorganisms have been producing them, and some other microorganisms have been living in the environment with antibiotics hundreds of millions of years, and the resistance mechanisms have evolved. So the agent resistance mechanisms might slowly be transferred to the clinically important part. But of course, if we are using antibiotics, then we are creating a selection pressure. So we need to understand that without selection pressure, you use antibiotic resistant mechanism have a fitness cost for the bacterium, and you will not have them. But if you are using somewhere antibiotics, then you get antibiotic resistance. And of course, people, if I say are talking about antibiotics, then we are using antibiotics in the hospitals on human patients, and this has been the main situation where we are talking about antibiotic resistance. But of course, humans are in contact with animals. So if you are making a Google search for pictures of dog and childs, this is what you are getting. We are in contact with the environment, and moreover, in addition to the pets, so the biggest problem is animal husbandry. This is a situation in the United States some years ago, but this hasn't changed. So if we use most of the antibiotics on the livestock, and this is parts that we are using on humans, and pretty much this chart is valid for the world already nowadays. And so this said, I would say that the European Union, for example, has taken some steps to decrease antibiotic consumption in animal husbandry, and we have had a decrease of 60% of general consumption during last 10 years. So for what antibiotics are used in animal husbandry, so one issue is to treat the diseases, and the other is also to use it for prevention. So that animals are packed so tightly that the disease is spreading. So you want to prevent. You say that okay, just keep the antibiotic for everybody, and then after the antibiotics were discovered, then it was found soon that if you give antibiotics to animals, they gain weight more rapidly. And from 1950s, the farmers started to keep the antibiotics at low concentration in the food as growth promoter. So this effect is occurring probably because change in the microbial communities in the gut. And the prevention and using as growth promoter is prohibited in the European Union nowadays, and that's good to see that we are decreasing antibiotic consumption and antibiotic resistance, but this is not the case for the world. So yeah, here you see the animal farm. So why the consumption has been so high is that the animals are packed tested together, and there is no point to treat one animal. So you need usually people have been using group therapy, so you meditate towards the farm, and how the antibiotic resistance is coming out from the farm is one is with the contact with the personnel. So this was a famous case in Netherlands, where in animal husbandry they started to really take action. Was that the same medicine resistant Staphylococcus aureus, the MRSA, the strain emerged in pig farming, and there was a farmer, and he went through, and his wife was a nurse, and the nurse was bringing the MRSA to the hospital, and we had hospital outbreak with the same strain. And this was one of the incidences that made the action, made the government in Netherlands to act. And of course, other contacts in the farm, and then from the farm it goes out with the food. So this is always antibiotic resistant bacteria. They are heat treated, so people have been testing in the slaughterhouse that in the morning the processing lines might be clean, but after this, you will have one animal with MRSA, for example, passing the slaughterhouse, and then the next products all contain MRSA. So the people from the food hygiene actually also raise the questions that what happens with the food in your kitchen that has been containing a lot of different bacteria that are antibiotic resistant and can be transferred. But of course, with meat, we have this is one issue, but certainly you have some manure, sludge go to the fields, and what is happening on the fields. What we also see in Estonia is that by the law, the material should not go to the water bodies, but if we are testing groundwater, then we actually see that actually the antibiotic residues and resistant bacteria go to the natural environment, to the waters. Specifically, you are not allowed to put sludge and manure on snow, but sometimes during the spring in Estonia, you put it on the soil, and then it snows, and then it melts, and then you have some amount of it in the rivers and lakes. So it goes downstream, and then in some loop it will meet with the environment for humans. And of course, sometimes we have food safety issues and trust of antibiotic resistant organisms also with vegetables that are eaten raw. So you see the onion, for example, and other vegetables. So if we're using antibiotics, then it spreads out from the production system. And of course, what happens to the antibiotic residues and antibiotic resistant bacteria in humans? This goes out through the kidneys, the urine, and then we have fecal materials that goes to the wastewater system, and the wastewater system actually is not built to remove the antibiotic residues and resistant bacteria from the system. So they are actually built to remove the nutrients that there are no eutrophic processes in the water. So they are not cleaning the water, and one thing that we see is that you will have antibiotic resistant bacteria from humans in those pools, and in some countries, actually in Sweden, we have some regions near Kalmar where you have a lot of water birds coming to feed here, and we are currently testing how the antibiotic resistance is being transferred with birds around the Baltic Sea. So and they actually transfer the antibiotic resistance out from the wastewater treatment plants, and also as so the water birds has been if you're looking at the natural environments, and the water organisms actually are transferring this risk factor for sure. So in the Americas, there is gulls that migrants during between North and South America, and this one was bringing antibiotic resistance from Chile to Canada. So and now we are looking what other processes around Baltic Sea, what we see in the birds. And we have been looking at all the segments, as I told, from this One Health perspective. So we have been working closely together with medical sector and with food and veterinary sector and with environment, and we have been looking at wastewater treatment plants. And with wastewater treatment plants, you can like ask the question how they can be more efficient with coming out from the wastewater treatment plant. And the other issue is always that can the wastewater treatment plant be a cheap diagnostics. If some material is coming in from a large amount of population, can we ask how much infection we have in this population? So we were doing it already for some time for antibiotic resistant bacteria from different populations, and then when the COVID-19 came, then we started a large testing campaign and releasing the news every week. And we continue. We just next year we continue with the wastewater surveillance of different pathogens. So now we have like I think 40 pathogens of this. That this is a European project where we can we want to see what we can test with wastewater. So certain antibiotic resistance, we have some markers, we know what is important, some viruses, and let's see what can be done. But maybe it's like an overview what is in Estonia. Like if you are asking where is Estonia stands from the viewpoint of antibiotic resistance. I think in general our situation is good, but still always you have to consider, I mean, really the issue is that people travel, as you see for infectious diseases. We have a lot of people traveling to other regions. If there are other regions in the world, people come back with antibiotic resistant infections. And also important to consider the goods, materials, agricultural products coming to us with large ships from Africa, America, Southeast Asia, where the resistance levels are high. So in general in Estonia, antibiotics are used at low level. This is both for human medicine and veterinary medicine. But in veterinary sector, the problem is that high proportion of third and fourth generation cephalosporins and fluoroquinolones. So those are the compounds that we really would reserve for human medicine, and better we don't want to see them in the animal husbandry, and it's even not sure why you have so high consumption of fluoroquinolones, but for third and fourth generation cephalosporins, we have these biggest users, their animal in the European Union. And what do you think, what has been the reasons that we use such big amount of cephalosporins? So what is issue with cephalosporins? It is a problem in human medicine, but the advantage in some parts of the veterinary medicine that they are unstable in the body. So if you have animals, then you don't need to throw away, especially in the meat industry, you don't need to throw away the meat. So so it gets degraded rapidly and you can use meat. So this was found in the our milk farmers was found that it's really nice to use the third and fourth generation of cephalosporins, and therefore it's so high. So we are trying to take it down. So let's hope that we can do this. And in human medicine, it's also good. What we found that during last 10 years, there is a transition to more advanced antibiotics without clinical need. So basically, beta-lactams, like simple penicillins, sometimes are effective, but people still do cephalosporins or penicillin with inhibitor and so on. So the physicians make the step to the more advanced, and it advertises to them and they think that, oh, it's more advanced, would be better, but they do this decision also in the clinical need. So that's what we observed. And in some cases, we have a lack of guidelines and implementation of the guidelines. That was also observed, but in general, in Estonia, the picture is quite good with some groups. Okay, so some questions about this part. So now I'm going more into really from antibiotic resistance and general questions to one of our topics on antibiotic action. Yeah.
Considering that you mentioned also yourself that okay, in Estonia the situation looks to be good. We are not consuming many antibiotics or a lot of antibiotics. But for example, today I was in the city center. I was walking there. There are a lot of Chinese tourists in Tartu. Those places that they use quite a lot of different antibiotics, and probably they are carrying all these different microbes that are resistant to bacteria. Would it make any difference that if we don't use what these ones are using?
Yeah, of course, I do. I mean, people travel, and actually, if you are looking like a worldwide perspective, is medical tourism is something that because if every people are traveling, that's not so big problem. But what we see is that if people go to the hospital in another country, like in France, they have their French-speaking previous colonies, and people from there sometimes, so quite often actually, if they have difficult diseases in France, and from that they get a lot of resistant infections, and France has a problem of this kind. So every people is not transferring so much. If you since we have been discussing not even with China, but we have the border with Russia, and the resistance levels in Russia are much higher than in Estonia. For example, if you are comparing the northeast of Estonia with St. Petersburg region, then there is a really big difference. So it has been not coming fluent to us, but certainly now I think that every time where there is war, then we have a problem with infectious diseases. So from Ukraine, we are going to get a lot of stuff, I would say. So, for example, multi-drug resistant tuberculosis is a problem. So if people don't have food and they are in shelters, it's wet, cold, and then they are refugees here, then certainly what you expect to have in the coming years also there. Yeah.
I see that you have proportion use of fluoroquinolone in veterinary sector, but on the other hand, in human medicine, in human practice, we know that fluoroquinolone resistant sometimes.
No, I mean, the idea was to have the critical medicines for human health out from the veterinary. So that's the idea, was because it is like what has been going with different layers. People first didn't think about this, and then with the veterinary use was already so high, then they thought that okay, maybe we should reserve those for human medicine. But the veterinary system already was used to use them. I think that the latest was with polythene and polymyxins in general, because it was found that they spread from because they were actually reserved first only for veterinary medicine. And then if there was a shortage of antibiotics to human medicine, then they gave a critical drug for the human medicine. And then people saw, especially in South China, when this was 2015, was a report that it can transfer so easily and so on, and it's a big problem that it comes from the animal husbandry, and then we want to increase rapidly now the polymyxin consumption in animal husbandry. And it's certainly, you see how sad situation is, that this is not certainly a drug that you want to use on humans, but if you don't have choice, then you use it. So basically, it kills the kids. So it's very toxic to them. But it's used for the critical case. So, and now we do what do we do in the lab. So this might be, so we want to use the current antibiotics mobile. Now we are coming to our question that usually we need to develop new antibiotics, but here you have the preclinical stages, and then you have the different phases of clinical trials, and your antibiotic can fail at any time, so it's really expensive. So it's not so easy to bring new antibiotics to the market. So we need to use current antibiotics less and maybe different. And that is something I hope you have been all been in microbiology laboratory classes, through bacteria. So you dilute your antimicrobial agent, and then add in the different wells or test tubes, you have the bacteria, and then you see how the antibiotics act on your bacteria, and then you see minimal inhibitory concentration where your antibiotics have some action. And the question is that for this test, you need bacterial growth. But in fact, not all bacteria, specifically if you have chronic infections, not all bacteria are growing. This issue was actually discussed already just after discovery of the first antibiotics. And this was Irish microbiologist Joseph Bigger, and just after the penicillin came out, so Bigger reported that penicillin cannot sterilize bacterial cultures. And what is happening in bacterial cultures, if you grow them, then actually, even if they are genetically uniform, they contain bacteria in different physiological stages. They are getting quite dense, a lot of bacteria together. They actually differentiate into different subpopulations. And if you treat them with antibiotics, any bacterial culture, what you can observe? First, resistance. This means that if the bacteria are resistant, then they can grow with presence of antibiotics. If there is rapid killing, like in green here, then they are rapidly killed. And then for antibiotics, we are also using the term tolerance in a specific way. That tolerance means that the killing is not so rapid. The bacteria are killed by the antibiotic more slowly. And what we usually have with the population is that we have this killing phase that is rather rapid, and then we have some smaller subpopulation that is refractory to antibiotic, and we call those ones persisters. So in general, disease is related to the tolerance and the persistence. I think the first is tuberculosis. Tuberculosis treatment, when it started, was streptomycin. It was more than a year. Now it is like 5 months with a combination of 4 or 5 drugs. Now we are getting a 5-drug combination with betactinib. So the issue is that the bacteria itself, if it's happy and grows and so on, it is affected by the antibiotics very rapidly. But you have those persistence hidden somewhere, and for this you need the treatment for months and months. Chronic urinary tract infections. So this is specifically a women problem. Men have prostatic problems in parallel. So you have the bacteria hiding into the epithelial cells. Cystic fibrosis, this affects the lungs. Pseudomonas aeruginosa usually is hidden there, and it's hard to get rid of it. Chronic infection of the joints, big problem of orthopedics. You put in the joint, and then with this foreign material, you will see bacterial growth, and if it's already there, then you maybe need to cut out the joint or something like this. It's really hard to joints, and specifically so prosthetic devices. Some antibiotics kill growing bacteria. So antibiotics can also like act in some acute phases, just being bacteriostatic, not allowing bacterial growth, but chronic infections we usually want them to be bactericidal. And our model organism as Escherichia coli, that is causing our strain has been causing chronic bladder infection. It actually is a problem that because of bladder, it can go in our animal models. We see in original patient, it was going to kidneys, then it goes to the kidneys, and the kidneys are dead, and then from kidneys it can also go to the bloodstream infection. So the urinary tract infection is not so mild as it usually is. So it can be a big problem to the kidneys, and it can lead to systemic infection. And then we were asking that if we are taking in our library, like we call library a collection of chemicals that are available for researchers, for different organizations. So we are taking a collection of chemicals that has been approved as medicines. This is like 1600 or something like this compounds, and cells are 4,000 or more, no more, close to 5,000. So we had compounds that have been in clinical trials. They might be not coming to medicine yet, so maybe have been abundant, but why you use those is that you already have some information about how they act in humans. What is the toxicity, how it's spread in human body, that is called pharmacokinetics and so on. So you have, if they are existing drugs, then it's much better that they are approved and we have some experience with them. Because if you have something new, you need to start from zero. So and this approach is called repurposing. So basically this is a full pipeline, and the problem repurposing actually is approaching only the last steps of this. So it's shorter for this development that you say that okay, you have been using the medicine for this indication, let's use it for something else. And now for our assay, you need to understand the bacterial growth curve. So if bacteria have a lot of food, there is small amount of them, then they start growth and they multiply logarithmically, and then if there is not enough food anymore, then they get into the stationary phase. They cannot multiply anymore, and there might be a death phase later. And now we are looking at the exponential phase. So we are having non-growing bacteria into this situation, treating them in the stationary phase with different parts, and then letting them grow again. And basically, we do it in a plate with many wells. So you are not doing it manually, it's all automatic. So you have a robot, a machine that is handling the bacteria. And then here are the controls, 2 antibiotics, antifloxacin and phenafloxacin. And here is just control that if you dilute the control, then they start growth more rapidly. So you treat with antibiotic, and then you dilute them into the fresh medium. And the antibiotic will not act anymore, but if the antibiotic acted on the non-growing bacteria, then the regrowth will be slow. Okay, so the treatment is during the stage when the bacteria are not growing, and then we put them in the fresh medium when they have the opportunity. The antibiotic is diluted out, and then they have the opportunity to start growing. And you can also make this as a one-point test. If you have thousands or later tens of thousands of compounds, then you just have one optical density measurement that, okay, you see that here something is happening. So that's growth is delayed, and then you can go back to those positions and analyze them in more yeah, good way. And basically, yes, we are measures that prove reduction time. This is basically giving the same information as on the previous slide. And then this was happening with our complete library. So here is controls that doesn't have any effect, and here are the two antibiotics that are supposed to kill the bacteria in the dormant state. And here we have some compounds that also delay the function. And then we did the tests first in E. coli, this uropathogenic strains, and then we compare are those ones that we see that have some activity, are they active also against other bacteria like Pseudomonas aeruginosa or Staphylococcus aureus. And then of course, if we are like having this kind of simple assays that we can make automatic in the systems, and we really like are looking really the ability to form colonies on the solid medium of the bacteria. Here we dilute just the medium, and here we go buffer, and here we dilute antibiotic, and we are getting the curve, the killing curve on the really live bacteria that we see. And here you see that the killing effect on different antibiotic concentration and then the effect on growth growth, and basically here we see that there are different groups who didn't find something that is not antibiotic or suspected to be antimicrobial, but certainly was interesting to find out three groups of antibiotics that really are sometimes effective against non-growing bacteria like rifamycins, macrolides, fluoroquinolones. And it's interesting, we are coming back to the anti-cancer drugs. And many of the compounds against E. coli, they were also active against Pseudomonas aeruginosa, less against Staphylococcus aureus, and it's expected because the cell wall is different. And this is really interesting with some of the compounds that people didn't expect that they are effective against Pseudomonas aeruginosa so much, like ciprofloxacin. And we see that ciprofloxacin is when we were measuring the effect against non-growing bacteria, then they kill non-growing bacteria quite efficiently at quite low concentration. So this concentration is something that we can easily achieve in human blood human treatment. And the most biggest surprise where we saw a reproducible effect is solithromycin. So if you know those drugs like erythromycin and glithromycin and so all macrolides, so this is a new, it's marketed from 2015 in US, and it is not supposed to kill bacteria. So it's by the textbook is bacteriostatic, and it should not be supposed to be active against Pseudomonas aeruginosa. And you see how strongly bactericidal it is against non-growing Pseudomonas aeruginosa. So we clearly have a new indication for solithromycin to try, because we, as I listed, like cystic fibrosis, it's really raised to the cystic fibrosis bacteria. It was the islets, all macrolides are actually food for the islets. And you can have, and we have some other really hard to treat chronic pseudomonas, which in most cases we get some ideas for trying and solithromycin is really interesting discoveries that we can be used. And here, this is if we are looking at pseudomonas, you will have this MIC. This is the effect on the growing bacteria, minimal inhibitory concentration. And here you will have the effective concentration of the regrowth event. And here you can have compounds with MIC like 10 or 100, and the killing effect is below 1 micron. So we really have this illustrates that we're really getting some compounds that if you are using the rule standards of the antibiotic discoveries that relies on the effect on MIC. In MIC, you say that it's absolutely hopeless, but for this assay, we will see that they have a strong effect in concentration that you can really achieve with during treatment. And then was also interesting, this is actually a proof of anti-cancer drugs. And the effects here are all in the range of like 1, 2.55 micromolar. And those are anti-cancer drugs that different physiological processes affecting the human cells and some DNA-related processes. And actually the concentrations where they are affecting, they are much lower than is used during cancer. So in the cancer treatments, they are quite toxic as the cancer drugs usually tend to be, but at those concentrations seem to be not. So if there is nothing that can be done, so those groups of anti-cancer drug should be actually investigated as a route of using treating chronic infections, and slowly actually we have already a couple of cases where some of the drugs have been tried, for example, chronic staphylococcus aureus infections, where especially in the joints where there is nothing else could be done. So we have a couple of reports, but as a group, I think this is we also found that point there can have a strategy. And then conclusions from this part. New tests for estimating antibiotic efficiency are needed. So if you build in the need to consider the minimal inhibitory concentration in your antibiotic development pathways, and you might miss some important compounds that are effective in some clinical settings. And pharmaceuticals, antibiotics can be purposed to treat different indications. So because usually if there is antibiotic development, then it's aimed, say, makes this target product profile in the company and they go to a small number of economically important indications that are not that easy to prove and test and prove, but you need to take different ways to approach other options. And then I would thank the collaborators for the second part. The first part had so many that I was also talking with more general way. Since we have been done during last 2 years from our lab, Eva Valtalo and one Perkins and we got help. It's a clean screening and it's equipment from Stina Berglund from and Vasilia Avelio from Sweden, from Mona. So thank you.
Do you do the screening in your lab or is it all happening in Sweden?
No, we did high throughput screens in our lab, in Sweden, but the smaller screens we made this like if we have so thick compounds and we do it ourselves.
You have the robots and you have.
Yeah, we did. Yeah, some kind of some level of robotization, but I hope that in some months or half a year we will have a large robot also here, so we can handle larger libraries like 10 or 20,000.
And why do you target particularly the bacteria at the time that they have reached to the somehow the stable growth?
Yeah.
Why not to at the start to?
That's the point that previously everybody has them in the start. And our question is that if you don't have them from the start, if you have a chronic infection, then you have bacteria that do not grow. So basically why you want to target doing some ones that are in the start and grow.
But would it not be telling that when you have at that concentrators at 24 hours, you're keeping them there, then when you dilute them back, put them back to you're actually having a single residuals of the medication there.
No, we dilute it enough, so it's considerably about below the MIC outside.
You are 100% sure that it had nothing to do with the gain to go to the lab, right?
Yeah, it doesn't look so. So basically, why we do it is just asking the question that what happens if things don't go fast.
It's somehow like the biofilm formation for the time like that, it's somehow depending on the different kind of culture that you do, you would have different properties and different somehow effects. This is one way of looking at that. And what do you, for example, you showed there are a number of compounds that are showing somehow interesting results. How fast you can take it to the clinic? You can immediately with this data, you can start to bring it to the patient?
In principle, you can. So this is always like the question that the physician always can use a drug that is approved. So and they can try it for different. So basically, if having the patient that really needs something. Then they can try it. So they cannot. So having it as a clinical trial, you need some approval and some more clinical work maybe. But in principle, you can use it. So we have been, for example, another macrolide is azithromycin. So with azithromycin, because for some of the drugs you have in this way you have some preclinical indication, and then you look how you go from the preclinical finding to the clinical finding. But with azithromycin, it happens the opposite. So Salmonella infections are actually really widespread in Estonia. It happens from time to time, and we take care of them, and it's not a major problem. Also, antibiotic resistant Salmonella It's coming here also, but it's a big problem, for example, Africa, South Asia, especially in Africa. So it's a really big burden of infection from the poisons, salmonella. And it's getting antibiotic resistant. And since they didn't have anything usual to treat it, and since it started with a macrolide azithromycin. So with azithromycin, so the minimal inhibitory concentration of the strains is also like 16 micrograms per ml or something like this. And the large concentration and of azithromycin is like 4. So you do an experiment. So people didn't expect that azithromycin is effective, but then people started to use it clinically, and it is clinically effective. So now we try to find out why azithromycin can treat salmonella. So basically, it happens in both ways. So the clinicians use something if there is no other option, and of course that's also the case that not for all bacterial infections you even have it in the trial. For example, the famous case is bacterial meningitis. Nobody ever made a clinical trial for bacterial meningitis, but people still treat it with antibiotics.
Question, especially the chronic infections in implanted devices. Like the chronic infections means probably the biofilm formation around the device. So when you repurpose the anti-cancer drugs to this kind of infections, is it the killing is probably, to me, I felt like it is because of their high penetrability. The anti-cancer drugs normally they have this more penetratability than the antibiotics. Maybe will that be the reason?
Might be,
that?
yeah, might be, yeah. We don't know. It's also here the question is, the next question is what is the mechanism that we say get better into the bacteria. Is there a lot of related uptake? And Sasa is that it's a biofilm, and especially in the prosthetic devices, it has first you get biofilm, then you have biofilm extracellular matrix, and then you will have inflammation, and the tissue by itself is making a capsule from the tissue, so it's a multi-layered thing, and cancer tissue will not give them a lot of blood. This means that also the drugs penetrate more easily. So I don't know about anti-cancer drug, about prosthetic devices. It's too far, but would be interesting to try because that's a big problem. Because if you finally need to, even in our hospital, I mean, if you need to cut it out and put a new one, that's hard surgery, and people have a lot of complications.
Yes, the other ones, especially we realize with your data, we realize that MIT is not the ideal value to detect the activity of antibiotics. So meaning that we have seen the upper and the lower levels around the MIC to check the effective lower.
You can have different assays if you have a different set of indications that is interesting for you. Then you can think of different assays. Because there are other things like this non-growing bacteria. What is another trend is several infections that people are thinking that are extracellular are actually intracellular in the mammalian cells. So people have been a lot of dispute actually salmonella infection now people claim that it's mainly intracellular, Pseudomonas, those intracellular, Staphylococcus aureus, those intracellular. So if you're talking about this prosthetic devices, and especially not even prosthetic devices, but we have a lot of chronic just joint infections. And there, Salmonella seems to be all in intracellular. And in intracellular, we have a different condition. This, I didn't have time, but we also need a different pH because also antibiotics, most of antibiotics are not effective at acidic pH. But the bacteria survive, and in those endocytic compartments, the bacteria in acidic pH. That's another issue. So if we are talking about this fluoroquinolone group again, this fluoxacillin cells, norfloxacin, and so on, ciprofloxacin and also fluoxacin. So there is a new ciprofloxacin, pinafloxacin that is specifically the indication of being intracellular.
I have a question.
Yeah.
Maybe I didn't notice, but there are so many diseases and so many drugs. Why did you use anti-cancer drugs?
No, we use those drugs. That's why I told we need this machinery, and this is what we got from 6,700 compounds. So we in the beginning we tested 6,700 compounds. So this is what came out.
Okay. And another question also. While you're speaking about non-growing bacteria, you meant persistence.
No, it's basically stationary. We are thinking now, is it applicable for persistence also? Because populations is always a really difficult, we always need to think. When we talk about persistence and so on, which kind of persistence, and that's really true that with those non-growing bacteria, you always have troubles at what kind of model system you use and what is relevant for this. So that's still a very deep open question.
Can you go back to the slide that you showed the growth of bacteria?
Growth curve?
Yeah. Yeah. Then in your, how you do it, you don't add your compounds at the beginning or so till they reach to the stationary.
Yeah, we treat it here.
Then 8 hours you are growing your bacteria without anything, and then at that phase you add these compounds to it.
Yes, and then we dilute it basically to this point.
To this.
I also have a small question, maybe I don't know much about bacteria, so it might be very simple. You know, in the mammalian cells, I know that we take up small molecules or liquid up from the external environment by endocytosis. Like there are different types of endocytosis. There's the machinery, the mammalian cells. In bacteria, I read like it's different. Endocytosis is not a machinery. And like how do you explain these antibiotics if they are to go inside the bacteria and destroy it? What is the possible mechanism, and is it different between gram-positive and negative bacteria?
Yeah, it certainly is. So this is the reason why you have. It's much more difficult to find new compounds from negative bacteria because they have two membranes, and the two membranes have different properties. So the outer membrane usually have general pores, and it's for many drugs, it is possible to go to support.
Spontaneously without
Yeah,
consequence.
yeah, and then they also diffuse through the inner membrane. But that a lot depends on the drug class. So we always have this question how much different barriers contribute. So, and it's also different in different bacteria. So for example, Pseudomonas is using certain amino acid transporters. You get in some antibiotics, but still some diffuse. So it's a big combination how they get in, but usually they get in, and also for they pass the membrane, but not all. Some of them need to go to the
So
core.
is it okay to say that it is more simpler in bacteria for molecules to pass through than mammalian? So there's no much barrier.
No, I think in the mammalian cells, there is also some of the drugs for intracellular targets, they just go to the membrane. They need to be having certain possibilities to get into cell. If they are very charged, then they cannot, so they can act only on the receptors and outside.
No, we have time.
How long did it take for you to do 6,000 different compounds in this assay system?
Well, it's first you need to set assay. Making it is not so difficult. It's maybe a week.
In a week, you can do 6,000 different compounds.
Yeah, so libraries are, because this is something that has been developed in drug development. So companies were collecting all those libraries, and first they found that they had a problem of quality of those libraries. If they get hits, so they cannot synthesize and repeat the results and so on. But companies made a lot of screening libraries for a lot of different diseases. So they identify a target that they want to inhibit or stimulate, or they were found important to cure the disease. That can be biochemical, enzymatic assay, some color assay, or room cell assays, a little bit more difficult, so you have some cellular reporter and so on. And companies have chemical collections that they can rapidly screen. They are more than millions of dogs.
And the same molecules that you have found, for example, in the stationary phase or in the chronicles are effective. Some of them you have. Have you tried to also check that if you started with the lag phase or that exponential phase, would they be also affected by that part?
Well, basically, MIC is this one. So it is blocking, but so basically this is a block. So for Pseudomonas, this is actually a classical test, and this is our test.
It was affected both at the lag phase and also at the.
Well, you see, MIC, the interesting thing is that you have some really effects. If MIC is higher, it means that it is less effective. So 10 to 100 micromolar, this is basically not effective at all. But the effective concentration for the non-growing cells is pretty low here. That like 1 micromolar or less. So this means that, yeah, we did.
Thank you very much. Oh, yeah.
Ah, ja, Is it European politics?
Yeah. Actually. Yeah. Thank you.
Thank you very much for joining with us, and Professor Tanel, thank you very much for your time and everything. We'll see you all in the next session. Thank you.
Yeah.